Welcome to Creating a Family, talk about infertility. I'm Dawn Davenport, your host and the director of Creating a Family, the organization, the nonprofit. And today we're going to be talking about genetic testing 101 for those going through IVF. We will be talking with Rachel Cady. She is a licensed certified genetic counselor and a lead laboratory genetic counselor for Cooper Genomics. We'll also be talking with Jenna Miller. She is a licensed certified genetic counselor and clinical science liaison with Cooper Genomics. Welcome, Rachel and Jenna, to Creating a Family. We're so happy to have you here.
Thanks for having us, Dawn.
Thanks for having us.
Okay, so as we were even talking about before we started recording, the terminology it's not confusing, but it is something that we've got to start off understanding the terms, and it's a little confusing because about mid 2017 we went by one set of terms, and then after that, slowly but surely, we adopted a new set, and we now have a current terminology. So pre-2017, the terms that were used were pre-implantation genetic testing, otherwise known as PGT. That was kind of the umbrella term. And under that, we had PGS, pre-implantation genetic screening, and PGD, pre-implantation genetic diagnosis. For a variety of reasons, including the fact that technology was progressing and more things were happening, and those terms were no longer as descriptive nor accurate as they should be. So slowly, as I said, slowly but surely, we have adopted new terminology that was introduced then. And the current terminology, PGS, is now pre-implantation genetic testing for aneuploidies, and that goes by PGTA. PGD is now two things: pre-implantation genetic testing for monogenic single gene disorders, PGTM, and pre-implantation genetic testing for chromosomal structural rearrangements, PGT-SR. This sounds a little confusing. Just know that the term we are including the earlier terminology because some of our audience may have heard that, but we're going to only be talking and focusing, of course, on the current terminology now. All right, Rachel, if you could start us off, what is PGTA?
Sure. So PGTA, as you mentioned, the A stands for aneuploidies. And the definition of aneuploidies is an extra or missing chromosome. So this technology is aimed at detecting whether there are any extra or missing chromosomes in the embryos. We know that there should be 2 copies of each chromosome. That would be considered a normal result. So anything that has aneuploidy would be considered an abnormal result for the PGTA testing.
Okay, so we're testing to see whether there is an extra or missing chromosome. And if there is an aneuploidy, an extra or missing chromosome, what does that mean to the pregnancy, to the fetus, or to the subsequent child if it is born?
Yeah, so the reason that any aneuploidy is considered an abnormal result for this testing is that we know that aneuploidy does lead to adverse outcomes. There can be a variety of different outcomes that we could see. Really, the 3 main things that we think about are a failed implantation or a miscarriage during that pregnancy. And then there are a select few that could make it to a full pregnancy and a baby born. But any babies born with a chromosome aneuploidy would typically be expected to have a physical and or intellectual difficulty. One of the more common conditions that people are generally familiar with is Down syndrome, which is caused by an extra copy of chromosome 21, also known as trisomy 21.
So usually, from what you're saying, if there is an aneuploidy, it's not a sustainable embryo. The embryo will either not implant or will not carry to term. But there are some um,
Mm-hmm.
that is there something that is there something that we know about those that the aneuploidies that would allow the pregnancy to continue versus miscarrying early on or failing to implant?
Yeah, so it does depend on the chromosome that's involved. Really, what this comes to is, from prenatal testing, back, even before PGT existed, what we were able to look at was ongoing pregnancies for women who went through diagnostic testing either during their pregnancy or if they had a baby that was born with some different problems and they did chromosome testing
Mm-hmm.
on that baby. So because of all of that, we know that there are these select few chromosomes that could ultimately make it to a baby, whereas there are other chromosomes where we really don't see any babies born if there was aneuploidy for that chromosome.
Okay.
So there is a specific subset that we know have been reported in live birth.
So how many chromosomes can be screened for?
With the PGTA technology, most of the PGTA technologies out there are able to screen for all 24 different chromosomes. So there are 23 pairs of chromosomes. The first 22 are named just 1 through 22, just by the numbers. And then that 23rd pair is your X and your Y chromosome. So that's why we say that there are 24 chromosomes in total, since the X and the Y are different. So the technologies can test for all of them.
Okay, excellent. Now, who should consider having, I mean, not everyone needs, I would assume, PGTA, but there are some who, for various reasons, would consider having this testing. So, who are the women who should consider having this testing, or couples?
Yeah, so really for any women or couples that are going through the IVF process, this is an option for them. There can be many different indications for why and when the PGTA might be specifically helpful, but really the bottom line is if you are going through IVF, this can still be helpful even if you don't have some of the other indications.
Mm-hmm.
So just it can generally help you increase your odds.
Mm-hmm.
But some specific indications that couples might consider this for would be if they do have a history of recurrent pregnancy loss. It's possible that some of those losses could be related to chromosome aneuploidy, and this testing may help. Or if they've had previous IVF failures where they have transferred embryos that just haven't implanted. Women who are of advanced maternal age, we know that as women get older, the chance for them to have a chromosome aneuploidy in their pregnancy can increase. So this is also something that can be helpful and mitigate that risk during their IVF process. If there is a history of an aneuploidy in a previous, child or pregnancy for that couple, it could just be some reassurance because since that is very, close to them having had that experience, this is a type of test that may help ease their anxieties going forward.
Mm-hmm.
It can also help with selecting a single embryo for transfer, whereas in the past, before PGTA was available, oftentimes multiple embryos might have been transferred to increase those chances of at least one implanting and resulting in a successful birth.
Mm-hmm.
But because we know that PGTA already helps with that, we can select single embryos and still have a better chance for that successful outcome.
Mm-hmm.
And also, sometimes there might be a family history of maybe a sex chromosome aneuploidy for the patient or partner themselves, where they might have an increased chance that their embryos or their pregnancy might have an extra or a missing sex chromosome. And this can help with that. Or there might be a family history of a condition in the family where either it is directly related to the sex chromosome, specifically we're thinking about the X chromosome,
Mm-hmm.
or it might just be a condition that we know is more prevalent in males or more prevalent in females. And by detecting the sex of the embryos with this technology, that can again help to reduce the risks for a specific condition to perhaps happen again in that family.
So in the latter, the sex-linked conditions, the reason that you would suggest that would be because you would be able to determine the gender. Obviously, if you're looking at all 24 chromosomes, you're certainly going to know the gender of the embryo.
Mm-hmm.
And so you can simply choose not to transfer the gender of which you are concerned about.
Exactly.
Okay.
And for some of the conditions, that are caused by a mutation on the X chromosome. We'll talk about PGTM later, but that can be an option for some families, but for some it may not be an option, whether that's financial or time-wise or whatever reason that might be. And if they choose not to do PGTM for whatever reason, this can kind of be maybe a backup option that's not directly looking at the condition, but can perhaps help to reduce that risk.
Okay, let me ask you a question. We've talked about advanced maternal age, and I think most everyone at this point knows that we have drummed it in. It's been drummed into us that as
Mhm.
women age, not only does their fertility decrease, but they also have an increased risk of having chromosomal abnormalities in their eggs. So does that mean that younger patients that this testing PGTA is really not needed for younger patients?
That's a really great question. I think this sometimes is a bit of a misconception among younger patients in that, hey, I'm not considered advanced maternal age myself. So, this PGTA testing isn't necessarily going to help me, or I don't need this because I don't have that increased risk for chromosome aneuploidy. But we do know that everybody has a baseline risk regardless of your age. So yes, this advanced maternal age, unfortunately, the connotation that it has, we do know that aneuploidy risks increase as mom's age increases, but this can still happen very much in a quote unquote younger patient. And oftentimes the way that it comes up is, there's a lot of surprise in, this percentage of my embryos might be expected to be normal or euploid versus abnormal or aneuploid. And, the question comes up, well, I'm young, my
Mhm.
friends are young, my family are young. I don't see all of these people having miscarriages or, failed pregnancies or babies born with a chromosome abnormality like Down syndrome. But we have to remember that we're testing embryos at such an early stage that we are picking up these chromosome abnormalities that otherwise wouldn't have been detected in, let's say, a spontaneous pregnancy. So we're really picking these things up before those adverse effects would have happened to that embryo.
It probably is helpful for us to talk about at what stage of embryonic development are we doing any of these genetic testings. And I don't
Mhm.
know whether Jenna, we haven't gotten you in. Would you like to address just the basics of when we're doing these testing? Obviously, it's pre-implantation genetic testing, so we know it's with embryos that have been created through IVF. But at what stage are we doing it?
Right. Thanks, Don. At this point, in this current period in time with IVF, we are typically testing embryos at what's called the blastocyst stage. That is somewhere between days 5 to 7 of embryonic development, typically 5 and 6. Some labs will test day 7 embryos, some will not, and that's just because those embryos took a little bit longer to get to that stage, so they may not be as healthy. But these days we're mostly testing those blastocysts,
Hmm.
and they have 100 200 cells, something in that range.
Yeah.
Earlier on, in IVF and PGTA, it was more common to test embryos on about day 3, when there
Hmm.
are about 8 cells. At that point we would take really typically only 1 cell out of the 8. Now when we're testing a blastocyst, we're taking about 5 cells.
Hmm.
um, 5 to 10 is the recommendation. Some places do more, some places do less, some labs, there are individual preferences there on precise size of biopsy, but it's typically done, again, on days 5 or 6, maybe 7.
Yeah. So, and actually, it's been a big shift, and it's been a big benefit from genetic testing to be able to wait, be able to take more, but that's somewhat dependent upon using, because it makes it more difficult to transfer fresh. But now that we have the research that indicates that, in fact, transferring after freezing and allowing the woman's body to uh, recalibrate after having taken the drugs for producing the eggs and having the egg retrieval, that there's benefits to that. So all of that has made this possible. Am I correct on that?
Absolutely. Yeah, vitrification, which is that fast freeze process for embryos, has really has enabled more individuals to do PGTA where they don't
Hmm.
have to be reliant on a fresh embryo transfer. It's also, like you said, helpful for, especially for women who are experiencing ovarian hyperstimulation syndrome, right, where it's just their body
Hmm.
isn't in a good place to transfer an embryo at that time. And one of the reasons why blastocyst biopsy and freezing has become more common is it was discovered that biopsying an embryo on day 3 versus at the blastocyst stage, day 5, 6, it showed that it had a more significant detrimental impact on the embryo at day 3 than when you biopsy it a little bit later.
Hmm.
So that's one of the big reasons why we've shifted to that later biopsy, and vitrification has just made that process easier and
Hmm.
less stressful when you know you don't have
Hmm.
to transfer right away and get results really quickly.
Exactly. Okay. Now, we've been talking about pre-implantation genetic testing for aneuploidies, PGTA. And we've talked some about who should do it, who should consider using it, and what chromosomes are screened for. But let's talk about more about what we mean about aneuploidy and what can the testing show us. So, Rachel, what can PGTA show us?
Yeah, so it is really important to think about what this testing can show us, as well as what it can't show us. So thinking about really the way that this testing was set up and designed is to pick up whole chromosomes that are extra missing, that aneuploidy that we talked about. But there are some other things that we can see, specifically. And I think Jenna will touch a little bit on this later, but we talk about segmental aneuploidies. So that is also something that we can detect. Jenna will talk about some of those limitations, but really what we're referring to with segmental aneuploidies is a smaller piece of a chromosome, maybe not the entire chromosome that's extra missing. So that's something that we could potentially see on a result. And we can also see some other types of chromosome abnormalities, such as polyploidy. Again, Jenna's going to talk a little bit more in detail about that, but polyploidy is something that we refer to when there's an example would be an entire extra whole set of chromosomes, or a whole missing set of chromosomes.
Yeah, I was going to ask. So we're talking about aneuploidies, which is well, euploidy. It means is a normal set of chromosomes. Aneuploidy means an extra or a missing. Yeah. So Jenna, what is polyploidy?
Yeah, so polyploidy is a chromosomal finding where you have an entire extra or missing set of chromosomes. So as Rachel said before, normally we have 46 total chromosomes, and we get 23 chromosomes from mom, 23 from dad. So one set from each parent.
Hmm.
But sometimes you can have an extra or missing whole set of chromosomes
Like you would be
from one
missing an
parent.
entire chromosome 5. Could just be missing.
So if you're missing an entire chromosome 5, that would be considered a monosomy. That's just one chromosome. We're saying chromosome,
Oh, gotcha.
that would be a missing chromosome or an extra chromosome 1, 2, 3, 4, 5, all of them.
Hmm.
You have an extra or missing. So in like the most common example that we typically talk about with polyploidy is triploidy. That's where there are 3 copies of every chromosome. So if you look in a triploid cell, instead of seeing 46 chromosomes, you'll see 69 chromosomes.
Oh, wow.
Yeah,
Okay.
triploidy is it's actually not all that uncommon. In humans, the most common cause of triploidy is thought to be when 2 sperm fertilize a single egg. There are other ways that could occur, but that's called dispermy, where 2 sperm
Hmm.
fertilize a single egg. Um,
Huh.
yeah, it's interesting. Actually, this
Yeah,
this
it
isn't.
is.
I find polyploidy very interesting. Unrelated to humans, this is just a fun fact. All of the seedless varieties of fruits that we eat are actually triploid as well. So like seedless watermelons, grapes, bananas, etc. They all have 3 copies of every chromosome because that makes them infertile, so they're not making seeds. In humans, having a whole extra or missing set of a chromosome is more detrimental than that. Typically, these things are not compatible with life. Triploidy in particular is a common cause of miscarriage. Occasionally, fetuses with triploidy can be live born, but they typically don't live very long. Hours is typically what we're talking about, minutes maybe even. And this is something that occurs in spontaneous pregnancies as well as in IVF pregnancies. It's not all that uncommon as just a general cause of miscarriage. And as we go on, we'll talk more about the technologies that we utilize for PGTA, but polyploidy is one of those findings that can be difficult for PGTA. You sometimes run into limitations here. For example, if you have a form of triploidy where there are, again, those 69 chromosomes, 3 sets of every chromosome, and the sex chromosomes are XXX, some technologies for PGTA can't see that, and while others can. So that's one of those areas where you can run into limitations.
Is this what you're talking about with we hear the term a balanced translocation. Rachel, what does that mean, and how is that detected, or can that be detected with PGTA?
Yeah, so balanced translocations are not the findings that we would expect to pick up on typical PGTA testing. Because the way that the PGTA testing is performed is looking at dosage or the amount of chromosome information that's present, a balanced translocation wouldn't be picked up because all of the chromosome information that's needed is there. It's just rearranged differently. So that's definitely one of the limitations for the PGTA testing.
Does that happen with segmental aneuploidy? Is that when you see balanced translocations where you've got a deletion of one chromosome that is added into another?
Sometimes if we're seeing patterns in the segmental aneuploidies of embryo results, it could be an indication that mom or dad actually has a balanced translocation. So if we're seeing, maybe we have a set of 10 embryos that were sent, or 10 biopsies that were sent for testing, and we're seeing that the same segmental aneuploidy is identified in multiple of those embryos, and potentially the reverse as well. So we might be seeing the same segment that is monosomy, and then that same segment that's trisomy in some different embryos. That could be an indication that one of the parents has this balanced translocation where they have all of the correct information, but because of the way balanced translocations work, it increases the chance that their children, or in this case embryos, can have unbalanced information, and that's where we see those segmentals.
Interesting. Okay. All right. So tell me a little about segmental aneuploidy. That's where we've got, it's generally smaller deletions or duplications. Isn't that right?
Yeah, correct. So they are smaller pieces of the chromosome, and with these, we can still see the same types of outcomes as we can with whole chromosome aneuploidy, like the failed implantation or the miscarriages. But because they are smaller pieces of the chromosomes, they may not be as lethal earlier on. So it is entirely possible for these segmental aneuploidies to result in a live birth. However, we would typically expect if it's something that's big enough that we're seeing on the PGTA technology, it probably has, a number of genes that are involved in that segment. So we are usually going to expect some type of physical or intellectual disabilities if that was to result in a child born.
Jenna, how effective is PGTA at detecting segmental aneuploidy?
That's a good question. It depends a little bit on the region of the genome that we're looking at. It depends on the what's called the coverage of the technology. When you're looking at various PGTA or any sort of genetic testing technology, there can sometimes be little gaps in the genome where the technology may not cover. And so if a segmental finding, particularly if it's a very small segmental finding, If it falls in one of those gaps, it won't be detected. For example, in our lab, we conservatively estimate that our resolution for our testing is about what we say 5 megabases. That means that we can detect segmental aneuploidies down to about 5 million base pairs or 5 million letters long. Some regions we can go smaller than that. Again, that's a conservative estimate, and it depends a little bit upon the coverage throughout the genome. But 5 million letters extra or missing is what we conservatively quote. And remember, for reference, there are 3 billion base pairs or 3 billion letters in the entire human genome.
Yeah, it does give us, it helps when you're talking millions, it seems big. So it does help to kind of put that in context of
Exactly.
what the entire genome is. Got it. Okay, perfect. All right. And this is something that Rachel, you referred to before about sometimes it's helpful, oftentimes it's helpful to be able to know from which parent, the egg or the sperm, the chromosomal abnormality originated. Why is it important, and is it possible with testing that we currently are doing to be able to tell which of the parents contributed the abnormality?
Yeah, sometimes this can be helpful. It certainly depends on the patient or the couple as to whether, they would want this information or use this information, but it can be helpful in some instances. The technologies that we have today, some of them can detect this, some of them cannot. So it does depend, and it's always an important thing to consider if that is information that you're wanting out of your PGTA testing. The scenarios where it can be helpful for families or couples going through with PGTA is especially if they're going through multiple cycles. If they're seeing that there is a larger number than is expected that are coming back as aneuploid, this type of information could potentially either help to reassure that, yes, this is what we were expecting versus maybe no, this is not, and it might change their next steps for their next cycle. And what I mean by that is, as we talked about earlier, we know that advanced maternal age is one of the bigger factors in a higher chance for chromosome aneuploidies. So if we have a family where they're going through a PGTA cycle and mom is of advanced maternal age, and we do this testing and we determine, yes, all or most of the aneuploid results were due to, they were from maternal origin. That does help to reassure that, yes, okay, this is because of the advanced maternal age factor, most likely.
Mm-hmm.
Whereas if we're seeing that it's reversed and maybe it's actually more paternal in origin, that there could be something going on that's just increasing the chance that, dad is contributing an extra or the missing chromosome in that case. And sometimes for families, they may just want the information to know and move on as they've been moving on before. But for others, they may want to consider other options for their next cycle, like perhaps a sperm donor or an egg donor in certain instances.
Mm-hmm. Yeah, so as to avoid the possibility. Okay. Now, we're reminding everybody we're talking about pre-implantation genetic testing for aneuploidies, PGTA. There are a couple of different testing platforms that you will hear about. And so, Jenna, can you tell us, there are two main ones. Can you just hit on the two main platforms that exist?
Sure. Yes, there are 2 main platforms that are currently in use for PGTA. There are a few others that have sort of come and gone as technology has improved over time, but the main 2 that are in use currently are next generation sequencing and SNP analysis, single nucleotide polymorphism analysis. So next generation sequencing is probably the one that most will hear about more often or be more familiar with. Next generation sequencing is used widely throughout all of genomics, not just in IVF and PGT. Basically, the way it works for PGTA is we're sequencing short fragments of DNA throughout the embryo's genome, and then we're just counting those fragments. It's kind of straightforward. So how many fragments of DNA are we seeing from each chromosome? So if we're seeing more fragments from a particular chromosome than we expected to see, then that would be a trisomy, an extra chromosome. Or if we're seeing fewer fragments from a chromosome than we expected, that would be a monosomy or a missing chromosome.
Mhm.
That's how next generation sequencing works. SNP analysis, again, that's single nucleotide polymorphisms. SNPs are just these single letter changes that occur every 400 to 500 base pairs or letters throughout our genome. They're very common. And for PGTA, we're looking for SNPs where you, at a particular SNP, you would either have an A or a B. You could be AA, AB, or BB. And we're looking for SNPs where you have an embryo that's AB, or it should be AB, and mom would be AA, dad would be BB, or vice versa. So basically,
Mhm.
we would expect the embryo to be heterozygous, have one A from one parent and one B from the other parent. If we're seeing an excess of ABs, that indicates an extra chromosome. If we're seeing a dearth of ABs, if we're only seeing one letter, right, that would indicate that a chromosome is missing or a piece of a chromosome. We actually use SNP arrays for PGTM as well. And I would recommend to, if you're undergoing PGTA, find out what technology is being used. It's just good to know. The different technologies have different positives and drawbacks. So, for example, NGS has really good resolution for mosaicism, but it's one of the technologies that struggles with some of those polyploidies that we talked about before. And versus SNP analysis is better for detecting those polyploidies and for detecting the parent of origin, like Rachel talked about, whether the abnormality originated in the sperm or in the egg. So some labs will use NGS, some will use SNPs. Actually, in our lab, we use both. We will utilize the next generation sequencing data to also look at the SNPs. And that way we kind of get the best of both worlds.
I was just thinking, it seems like most well-rounded would be, if possible, to do both, because that way you're making up for the deficits in each.
Exactly.
Yeah.
It really helps to be able to do both.
When you raise mosaicism, you raised a point that we get a lot of questions about. So I'm glad you raised it. It's a great segue. What is, and Jenna, I'll direct this to you. First of all, let's just say, what is mosaicism?
Sure. So mosaicism is a common occurrence, again, in all of genomics. This isn't unique to IVF or PGT, but in general, mosaicism is any time you have two distinct populations of cells with different genotypes, with different genetic makeups in the same individual. So in embryos, when we're talking about a mosaic embryo, typically that means that we're seeing a mix of chromosomally normal and abnormal cells in that embryo. And this finding is not related to maternal age, unlike whole chromosome aneuploidy, where we see that increase with advancing maternal age. This is not due to maternal age, and that's primarily because these sorts of mosaic findings are due to errors in cell division after fertilization has occurred. So when we're seeing age-related aneuploidy, that's related
Hmm.
to sort of the formation of typically the egg
Hmm.
before it's fertilized. But
Hmm.
mosaicism is due to errors in cell division post-fertilization. And those errors could mean that you started with a chromosomally normal embryo that acquired an abnormal cell line, or the opposite could also be true, where you started with a chromosomally abnormal, an aneuploid embryo, and that aneuploidy sort of went away in a particular cell line, and then you're left with a euploid cell line instead.
Let me ask a question. Back in the day when we were testing 3-day-old embryos,
Hmm.
then And you were only taking one cell. Was there a higher incident of mosaicism when you were looking at just one cell, or do you see a higher incident now when you're taking, any 8 to 10 or whatever number of cells at a 5 to 7 day old or 5 to 6 day old embryo? Is there any difference in the presence of mosaicism depending on how many cells you have biopsied?
There is. That's a great question, Don. So if you're only taking one cell from an embryo, you're not going to be able to see mosaicism because all you have is the makeup of that
Oh,
one
that's
cell.
yeah,
You need more
yeah,
than one
yeah.
cell to be able to see mosaicism. However, very early on in PGTA, when we were biopsying day 3 embryos, they would sometimes take more than one cell before they realized that's really not a good thing to do.
Mhm.
And they actually saw a higher rate of mosaicism in those day 3 embryos than we currently now see at the blastocyst stage.
We get a lot of questions about if you have mosaicism in an embryo, will it self-correct? Is it something that, and this may be outside of your area of expertise, but it is a question we get a lot. So feel free to say, this is not my area, but if it is, will it self-correct as time goes on and the embryo develops?
So that gets at sort of there are two different things to address there. First of all, this idea of self-correction, I personally don't love that term because it
Hmm.
implies some sort of sentience or some like intentionality there. Um,
True.
is it possible that there can be an embryo with a euploid and an aneuploid cell line, and the aneuploid cell line goes away or isn't still present, at later stages of development? Yes, that's possible, but it's not so deliberate. It could be that the aneuploid cell line, that the cells don't have the correct number of chromosomes, so maybe they don't grow as well. Maybe those cells are more likely to die, and then all that's left would be euploid cells. Some have suggested that those aneuploid cell lines in mosaic embryos are more likely to be preferentially, they're more likely to survive in the trophectoderm of the embryo than the inner cell mass. Now we're getting into some deep embryology here, but the
Well,
trophectoderm.
just but briefly, what we are biopsying is the outer cell mass, not the inner cell mass. Yeah.
Exactly, exactly. So typically we're biopsying the trophectoderm. That's that outer layer on the outside of the embryo. And some have suggested that those aneuploid cell lines are more likely to persist there than in the inner cell mass that goes on to become the baby. And
Hmm.
versus the trophectoderm goes on to become the placenta. And in a way that may be reassuring to some to think, well, it's less likely to be present in the fetus, although we
Hmm.
still want a healthy placenta during
I
pregnancy
was just
as
going to say,
well.
you also, yeah, the placenta is awfully important. So yes, we
Now,
still want a healthy placenta.
mosaicism is a very new topic that we're really just starting to get a grasp on. Before next generation sequencing, we really weren't consistently seeing mosaicism. So a lot of this is new and a lot of this is speculation,
Hmm.
or, based on very limited data. So take everything about mosaicism with a grain of salt.
Hmm. Are there different degrees of mosaicism? Like a lot of you see a lot of mosaicism, or just a little? Are there different degrees as far as the importance of what it means for the health of the embryo?
Yes, there are different degrees. So most PGT labs are separating mosaic results into two categories. They'll call low-level mosaics versus high-level mosaics. And the precise definitions of low versus high-level mosaicism, it differs by lab, and what the lab is comfortable reporting. So a PGTA result, it sort of falls along a spectrum of euploidy versus aneuploidy, normal versus abnormal. Most embryos are clearly one or the other, clearly euploid, clearly aneuploid. But when you have a mosaic embryo, it's going to fall somewhere in between, somewhere in the middle of that spectrum. And every lab has to decide where in that spectrum, where they want to set their cutoffs between euploid, aneuploid, and mosaic. So for example, in our lab, we define mosaic embryos as those where the signal we're seeing comes back between 80 and 20% aneuploid. And there are a few reasons for that. Some professional society guidelines suggest those cutoffs, et cetera. But the way that I think it's easiest to conceptualize this is by thinking about that typical 5-cell embryo biopsy. So if we're taking 5 cells from an embryo, and 1 out of those 5 cells is aneuploid, the other 4 are euploid, that embryo would be 20% mosaic, right? 20% of the
Hmm.
cells were aneuploid.
Hmm.
So if we're seeing a signal that's less than 20%, that would mean that it's less than one cell that's aneuploid, and that doesn't really make sense, right?
Mm-hmm.
And then that's also true on the opposite side. So if 4 out of the 5 cells are aneuploid, then that would be 80% aneuploidy. And if it's more than 80% out of that 5-cell biopsy, that would be more than 4 cells. So anything above 80%, we're going to call aneuploid. So
Mm-hmm.
that's generally where we get those 20 to 80% cutoffs for mosaicism. And then we separate the low-level mosaics from the high-level mosaics at 40%. So that would be 20 to 40% of aneuploidy coming from a particular embryo. We would call that embryo low-level mosaic, and 40 to 80% aneuploidy, we would call that high-level mosaic. But again, every lab sets their ranges differently. Some call mosaics at, you know, their total amount that they'll call mosaic is between 40 and 60% or 30 and 70%. The
Mm-hmm.
cutoff between low and high may be different. They may set that at 40 like we do, or at 50 or somewhere else. So again, this is something where it's good to know what lab you're using and what they do before you begin an IVF with PGT cycle.
So obviously, the reason we're talking about mosaicism is because ultimately it's going to have an impact either on the pregnancy, the fetus, or the baby. So does mosaicism impact implantation rate, miscarriage rate, and birth defect rate? And does it matter the degree of mosaicism? As is it directly corollary between the higher levels of mosaicism have higher levels of impact on those three things: implantation, miscarriage, and birth defects?
Yeah, absolutely. This is a huge question in assisted reproductive technology right now, Dawn,
Mm-hmm.
among scientists. But again, so the first report of babies born from mosaic embryo transfers was in 2015. So our research is somewhat limited, but the research that we do have is consistently shows that mosaic embryos implant less often than euploid embryos. They miscarry more often, and they're less likely to result in a live birth than euploid embryos. There's some research that suggests that low-level mosaics, where there's a lower proportion of cells in the biopsy, are aneuploid. There's some evidence that those low-level mosaics are more likely to result in an ongoing pregnancy and a live birth than the high-level mosaics. Actually, a lot of IVF clinics will only transfer low-level mosaics for that reason.
Mm-hmm.
But then some other research hasn't seen that difference. So it's still a little bit
Yeah,
in the
it's all a new field. We have not had the ability to even recognize or detect. So the
exactly.
information, we're so new. And tell me if I'm correct, but generally if you've got enough embryos, if a woman or a couple has enough embryos, almost always the choice is going to be to transfer one that is not mosaic. Let's say they have a mix of mosaic embryos and non-mosaic embryos. If they have the choice, most often they will choose to not run the risk, and they will transfer the euploid embryos, not the mosaic embryo. But sometimes, not infrequently, that we see that they either only have mosaics or they only have one and it's mosaic. So that's when the problem, that's when the decisions have to be made.
Exactly. Virtually everyone recommends prioritizing euploid embryos over everything else.
Hmm.
With these mosaic embryos, while they do, result in babies less often, they do result in babies,
Hmm.
and so their potential is not zero. And there's still a lot in the scientific community. We're really grappling with how do we properly rank these at a more granular level. Actually, just this week, they're at the European Human Reproductive Society conference at ESHRE. There
I was going
was
to say ESHRA.
ESHRE, yes. I'm like, what
I
do
don't.
all those letters stand for?
I know. I was thinking I couldn't even tell you. I'd have to see it written. Yeah.
Yeah. So at ESHRE this week, there was an abstract presented on the outcomes of 1,000 mosaic embryos transferred. And these were from multiple countries. There were a couple centers in the US represented in that, as well as a couple in Europe and one in Asia. And they, based on their results, have come up with, again, this is still preliminary, but they came up with a more granular scoring system. Obviously, the large categories remain the same. Transfer a euploid embryo first, then a mosaic, then an well, most say don't transfer an aneuploid, but they're at the end of the ranking there. But then there's still a little bit of question as to like what to do with the different types of mosaics, like segmental mosaics, for example, which that particular study saw did better than the other types, or if there's just one chromosome that's mosaic versus if you have two chromosomes that are involved in mosaicism versus three. And Don, I did want to also address the question that you had about the birth defect rate with mosaic embryos. So again, research is super limited on this, but to date, there have been no reports of babies born with birth defects after mosaic embryo transfer. That doesn't mean that it hasn't happened. It just hasn't been reported in the literature, or adverse outcomes aren't clearly related to the mosaic finding, because even a perfect embryo can result in a birth defect, regardless of how much genetic testing you do before or during pregnancy. There's always going to be a 2 to 3% background risk for birth defects. Some may even say a little bit higher. So as of yet, we haven't seen any indication of an increased chance for that. That doesn't mean it's not possible. And we do know that some of these mosaic findings that we see in embryos that have been transferred, we have seen in like the pediatric genetics community, we have seen children who carry those chromosome abnormalities who have birth defects. But as for when we see it in an embryo and then transfer it, there's no correlation to that yet. I will say that there was just a few months ago, there was the first report of a baby born following mosaic transfer where they did see that same mosaic finding carried through to pregnancy and to live birth. This baby was born in Turkey, and at birth there was 2% mosaicism for the same finding that was seen in the embryo, seen in the baby, but that baby had a normal pregnancy and was born totally healthy.
And I would be interested in seeing the research continue following birth. Number one, testing, genetic testing the child to see whether the mosaicism continued. And then also I'd be curious to see what at 5 years, 10 years, 15 years, 30 years out, if there have been an increase in health issues. But that data simply isn't available.
Right, and it won't be. Again, 2015 was the first report of babies born, and that's only
Right.
5 years later. So,
Yeah,
and
yeah,
that
exactly.
was a very small sample size. So it'll probably
Yeah,
take years, maybe even decades, to get that
right.
amount of information. But Don, you're absolutely right. I am just itching for that data as well.
Yeah. Yeah. I'm hoping when I was hearing about this 1,000, I'm hoping they get funding to continue the research every year on these now children who have been born. So yes, let us fund our medical research. We need it. All right. So now let's talk about what is the research currently showing about the impact of PGTA on the success rate of IVF, in vitro fertilization. Is it effective at making it more likely that the embryo transferred will implant and grow for 9 months into a baby?
Yeah, so PGTA has been seen to improve the pregnancy and live birth rate per embryo transfer. And this is especially true in women over 35, as we've discussed. The data for women under 35 is more mixed, although, as Rachel said, there are other potential reasons why PGTA could be useful in the younger population. But one thing that I like to stress is that PGTA is not going to change the cumulative pregnancy rate, meaning if you have a certain cohort of embryos that you got from an IVF cycle, PGTA doesn't change the genetic makeup of those embryos, right? So whether or not you use PGTA, if you put all those embryos back, eventually you'll find the euploid one. So let's say we have like 4 embryos, and maybe of those 4 embryos, 1 was euploid, 1 was mosaic, and 2 were aneuploid. If you're doing PGTA, you would transfer the euploid embryo first, the one that has the best chance of resulting in success, right? If you're not doing PGTA, you might choose between those based on morphology or other criteria, and so you may or may not choose the euploid embryo first. And so PGTA helps if you know you're transferring a euploid embryo per embryo transfer, you're more likely to be successful.
But in the example you gave, if you transfer all 4 embryos, one would, and let's assume that you get one pregnancy and it is from the euploid,
Mhm.
then you've transferred all four, but your cumulative pregnancy rate would be the same because you would have had one pregnancy out of this egg retrieval.
Exactly.
Yeah.
And again, especially in those women who are over 35 who are more likely to make those aneuploid embryos, it can be really helpful. And it can, in anyone, just decrease the time to pregnancy,
Hmm.
which is sort of what we're talking about there, right? You get to that euploid embryo sooner.
Hmm.
I believe Rachel also mentioned that PGTA has been shown to decrease the risk of miscarriage, just in general, not even in IVF, just in all pregnancies. About 50% of first trimester miscarriages are chromosomally abnormal, and that's the reason for the loss. So if you have a good indication that the embryo you're putting back is euploid, then that is going to decrease the chance that you will suffer a miscarriage because of chromosome abnormalities, right? But you do also have to think about how you want to define success. So one interesting finding about PGTA is that it's really increased the rate of single embryo transfer, and therefore it's decreased the rate of twins born from
Hmm.
IVF. So I know a lot of patients like the idea of twins, especially when you want multiple children and it seems efficient, and if it's been a long time and you wanted multiple children by now, I totally get that. But from a medical point of view, twin pregnancies are considered high risk, and
And they
therefore,
are high risk. That we just, yeah,
yes.
thank goodness we have reached the point now where I think that message has gotten through. On an emotional level, a lot of people still say, "Oh, twins," but I think we have gotten the message through that the risk, if you have a choice, you would not choose to put your fetus and baby through the potential for cerebral palsy, brain bleeds, and all the things that can happen with multiple births.
Right. So in the medical community, our goal is one baby per pregnancy. That's considered ideal and safest. And just at the American Society for Reproductive Medicine meeting in 2019, there was a big celebration about how much the multiple pregnancy rate has dropped in
Mm.
recent years, and that's largely attributed to PGTA. Because if you again have a good indication that an embryo is euploid, then you can be that much more confident just transferring
Mhm.
one at a time. Also,
Mhm.
vitrification helps with that, as you said before,
Yeah,
where you can save those embryos for later transfers.
right.
You don't have to put all your eggs in one basket, so to
Yes,
speak.
so to speak. Pardon the pun. All the embryos in one uterus, so to speak. Yes. Okay. So, what about the risk of false negatives and false positives? I mean, that's everyone's fear that they get test results back, and can they trust them? Because they're going to have to make huge decisions based on these test results.
Right. So false negatives and false positives are possible with PGTA. It used to be called PGS, pre-implantation genetic screening, because it's not considered a diagnostic test. It's considered a screening test, and it's a way of detecting the embryos that are most likely to result in success with IVF and embryo transfer, right?
Hmm.
Um, in our lab, we quote about a 97% accuracy rate with PGTA. It's difficult to calculate precise false negatives and false positives because when we're doing PGTA, we're not putting back aneuploid embryos typically.
Hmm.
Um, although there was a really interesting study done recently where PGTA was performed on embryos but then not assessed, not looked at until after embryos were already transferred without that information. And in that study, they identified that they had transferred 95 aneuploid embryos unwittingly, and none of those embryos made babies. A lot of them made pregnancies, but those pregnancies didn't make it
Hmm.
to term.
Right.
So the risks of false negatives and false positives are low, but not impossible. There have also been rebiopsy studies that have looked at this, where maybe an embryo has been donated, maybe it's an aneuploid embryo or mosaic, or even some euploids, where we then go and retest it, or we test the inner cell mass, right? That part that becomes the baby. That's what we really want to know, is what we really want to know what's going on there. And what we've seen is that if an embryo was called euploid from the trophectoderm biopsy, the likelihood that the inner cell mass is totally euploid is very high. We're
Mhm.
talking, high 90s%. And the same is true if the original trophectoderm biopsy was totally aneuploid. The likelihood of there being some degree of aneuploid cells in the inner cell mass is also similarly very high. Now, it could be a different chromosome is the issue in the inner cell mass, or it could be you saw an extra chromosome in the biopsy and you see that same chromosome missing in the inner cell mass. So it's not always exactly the same issue, but highly likely that there's some degree of chromosome abnormality in that inner cell mass. Now, where it becomes a little fuzzier is with these mosaic embryos. If you see mosaicism on a trophectoderm biopsy from an embryo, the inner cell mass could be euploid, aneuploid, or mosaic. And mosaicism is thought to be the primary reason for false positives and false negatives on PGTA. Remember, with PGTA, we're making an assumption. We're assuming that the cells that we took from the biopsy, we're assuming that those are representative of the rest of the embryo. And as we've just discussed, the vast majority of the time that's true, but occasionally it's not. And that's primarily thought to be due to that mosaicism.
Okay, that makes sense. All right, we've been talking about pre-implantation genetic testing for aneuploidies, PGTA. Now we're going to move to talking about pre-implantation genetic testing for monogenic single gene disorders. And that goes by the acronym, because of course we have to have acronyms, PGTM. But in this case, an acronym is good because there's no way we would we'd be able to say pre-implantation genetic testing for monogenic single gene disorders very easily. Doesn't roll off the tongue. All right, so Jenna, let's stick with you. So what is PGTM? What is it looking for, and who? Actually, Rachel, we haven't talked to you for a bit. Let me get you and talk a little bit about the PGTM.
Yeah, so PGTM, like you said, monogenic or single gene disorder. So what this testing is doing is looking at genetic conditions that are caused by a single change in a single gene, typically. So examples of this could be cystic fibrosis or sickle cell disease or Huntington disease. These are some common things that people might have heard about. And really this testing is looking to determine whether an embryo has inherited a specific mutation for that condition that we know of in the family, or if it has not inherited that condition.
So you know in advance what you are looking for when you're doing PGTM.
We do. We actually have to know in advance for PGTM. So this is a very specific test that is going to be tailored case by case. So for families that know that they have a specific mutation in a gene, specific condition in their family for which somebody has had testing and identified that genetic cause, this is the testing that we would be able to do to look for that.
Okay, who should consider having PGTM?
So really anybody that has a known genetic cause for a condition in their family can consider PGTM testing. Most of the conditions that we deal with are conditions that cause significant health concerns in either children, or sometimes these could be later adult onset types of conditions. But really these families can find out about this risk based on different factors. So sometimes they might have a family history of the condition in question, and the family member who has that condition has gone through genetic testing and identified that specific cause. And then the couple that is looking to build their family, has identified that they, either one member of the couple or both, do have that genetic risk. Mm-hmm. Sometimes there may not be a family history of the condition, though, and this often happens with what we consider autosomal recessive or X-linked conditions, where there may not be a family history, but perhaps the patient or the spouse have had genetic screening done in preparation for building their family, and they've identified that they carry a mutation, but they don't have the condition themselves. However, that is passed on in a certain way to their future children, that condition could be present in their child. Mm-hmm. So really anybody who, has this known genetic cause can consider the PGTM testing to look for that in the embryos.
And if someone were to choose not to go through, they have a genetic condition, or they're aware of it through their family, in their family history, or they found out about it through genetic testing, carrier testing. The other alternative would be to just go ahead and if they have no fertility issues, just conceive naturally, or even conceive through IVF without doing the genetic testing, and then test during pregnancy, either CVS or
Mhm.
amniocentesis. And can it be picked up that way as well?
Mhm. Absolutely. So that is a very common alternative option for families who decide not to go through PGTM, for which can be for a variety of different reasons. And the diagnostic tests during pregnancy, like the CVS or the amniocentesis, can detect these types of conditions. The difficulty with that is then the family can potentially in the position during the pregnancy to
Mhm.
decide if it is determined to be affected with that condition. What do they
Mhm.
do from there? Do they
Mhm.
continue that pregnancy knowing that they'll have a child born with that condition, or do they
Mhm.
potentially elect to terminate that pregnancy?
Mhm.
So while that may work for some families, typically the type of population that we end up getting for PGTM are the families that really can't see themselves going through that type of decision process. And by doing the PGTM, this helps to really reduce that chance that they would be in a pregnancy that is determined to be affected.
Mhm. That makes sense. And does PGTM impact the success rate of IVF?
It's not something that we typically expect to be related to the success of IVF
Mhm.
in
Because it really
general,
wouldn't impact the
because,
implantation or even early miscarriage, right?
yeah,
Yeah. Mhm.
exactly. There are a few different conditions for which we might test with PGTM that can be lethal early on in a pregnancy, or potentially have associations with likelihood of implanting or not. But the majority of them are not usually related to that. So yes, if it is a condition that we know is lethal during a pregnancy, in that sense, you could kind of
Mhm.
make that argument.
But that's not the
But for the
purpose.
most part, these are unrelated. Exactly,
Yeah.
it's not the purpose.
Mhm.
But what we can do for couples who are going through PGTM, we can add the PGTA to that. So they can do both tests on one embryo biopsy sample, and then you would of course get all of the normal benefits of the PGTA along with the information that you would get from the PGTM.
Okay, and so the timing of the biopsy would be the same as at PGTA. Is the
Mhm.
testing process itself, well, obviously it's going to be different because you're testing for something different.
Mhm.
But is it significantly different? Is it your test results faster, slower, whatever? How does the process itself, the testing process itself, differ from PGT?
Yeah, so obviously, like you said, we're testing something different. So in that sense, it's different. But ultimately, it is a really big difference in terms of the time and the steps that are involved in this process. So PGTA is a much more straightforward type of testing where there's really not much work that needs to go into it ahead of time. Patients can go through their IVF cycle, send samples to the lab, and do their test and get their results. With PGTM, we need a lot more preparation. So like we talked about before, we always need to know what is the gene and mutation that we're going to be looking for. So the first thing we always need are going to be genetic test reports for the family member or members that have had testing and have this known mutation. And based on that genetic test report, the lab is going to assess whether that is something that they can feasibly look for using PGTM or not. And oftentimes we will look to involve additional family members other than just the patient and the partner. So sometimes children or parents are helpful in this process, because ultimately what
Mhm.
we need to do with PGTM is to set up or design a test that's tailored to that family's DNA. So a lot of the process in the beginning is collecting information and reports. And then once we have that and we know what our plan is, then we need to go in and actually develop that test, which can take several weeks. All of this really needs to happen before the couple creates those embryos, because we want to have that test ready before those samples are sent. So that way we have something to use to test those samples.
That
So
makes
really
sense.
the time commitment and the steps are a big difference there.
How accurate, and one would assume the cost would be also higher. I mean, just because of the amount of time that you're talking about.
Mm-hmm.
How accurate are the results? At this point, you're banking a lot on the fact that you're hoping not to transfer an embryo that's going to have, in some cases, a fatal condition, and others life altering. So how accurate are the results?
Yeah, so the accuracy for PGTM, and again, this can certainly vary from lab to lab. Generally speaking, we are going to expect that accuracy is at least in the mid 90s or higher. So specifically for our laboratory, we quote at least 95% or higher for this accuracy. And, similar to what Jenna was talking about before with PGTA, there is that chance for false positives or false negatives, although, again, those chances are still expected to be relatively low. But going back to all of the terminology and everything, we don't say that this is a diagnostic test. It still is pre-implantation genetic testing, not an actual diagnostic test. So there is always that recommendation for follow-up testing during pregnancy as a confirmation.
And that would be the case with all type of genetic testing, wouldn't it? That we still recommend the standard prenatal testings that would be done on a pregnancy that was conceived without IVF, without genetic testing.
Correct.
Okay.
Correct.
Excellent.
And it's always a good idea to, have copies of your PGTA, PGTM, whatever testing you've done. It's a great idea to have copies of those reports to, show your OBGYN so that they're aware that there has been testing done, but also that, again, this was not diagnostic testing. It was screening with very high accuracy, but keeping that in mind as they're going through that, the process of the options for testing.
Okay, so now we've hit on PGTA and PGTM. So we have left PGTSR, which is pre-implantation genetic testing for chromosomal structural rearrangements. Again, an acronym is a blessing in this case, PGTSR. So Rachel, what is PGTSR?
So PGTSR is the technology that we use to identify essentially unbalanced forms of any types of chromosome rearrangement. So way back when we were talking about some of the limitations of PGTA testing, where we can't detect those balanced rearrangements that a parent might carry, this is where that comes into play. So if a parent has some type of chromosome rearrangement, whether that's a translocation, which is where essentially two pieces of two different chromosomes have switched places with each other, or an inversion is another example. An inversion is basically a piece of a chromosome that, if you kind of think about taking a little piece of a chromosome, pulling it out, turning it upside down and putting it back in. That would be an inversion. So these are some common examples of chromosome rearrangements where mom or dad might carry this balanced form. They've got all of the genetic information they need. It's just rearranged differently. So they are perfectly healthy. They don't have any health concerns related to that. But in their embryos, there is a much higher chance that translocation or inversion or whatever chromosome abnormality can become unbalanced, which then results in, like we talked about earlier, those segmental aneuploidies, pieces of chromosomes that may be missing or extra as a result of this. And so the PGTSR technology is aimed at identifying those unbalanced forms in the embryos.
Okay. All right. That makes sense. Now, so who should consider having this testing? How would you know that that you're a carrier of a balanced, some form of translocation?
So some people don't know that they are, and they might go through, let's say, the PGTA process, and we might by chance see some weird patterns that are suggestive that a rearrangement might be going on in a parent. And then that parent or those parents, mom and dad, might go on to have their chromosomes, analyzed to determine if there is a rearrangement that can be detected. Other times, they may find out before going through any IVF process, and maybe that's why they presented to IVF in the first place, because a lot of these cases, we're going to see that there have been multiple miscarriages. Again, just thinking about what we've talked about with aneuploidy in general, and of course, even the segmental aneuploidies, is that when you have extra or missing genetic information, one of those adverse outcomes is failed implantations or miscarriages. So for some families, they may have experienced multiple pregnancies that have miscarried, or they're even having trouble getting pregnant. And one of the tests that can be offered to those families is to have chromosome analysis done on the patient or the partner to see if they carry this chromosome, if they carry a chromosome rearrangement. So those are kind of the two most common ways that people might find out about this. For some people, they might have found out because, their parent experienced difficulties, and, they might have been tested when they were a fetus through an amniocentesis. But once you do know that information, you have had your chromosomes analyzed and determined that you do carry this rearrangement, PGT-SR technology is really the way to look at any potential unbalanced forms in those embryos.
Okay.
And then hopefully this will mitigate, again, that risk for those miscarriages or failed implantations moving forward.
And the reason, if you have reason to believe that you do have one of these chromosomal abnormalities or translocations, the reason you would do PGT-SR rather than just PGT-A is because PGT-A has a higher likelihood of missing it. Is that correct?
Yes. So what it really comes down to is the fine-tune review that goes into it. And again, this can vary from lab to lab, but a lot of the labs are going to use the same actual technology platform with PGT-A and PGT-SR, because again, that goal is to assess the amount of chromosome information. So that part is usually pretty similar, if not the same, but there is this additional level of review that goes into it, because with a PGT-SR case, we will know ahead of time that, this patient carries a translocation between this chromosome and this chromosome at this break point and this break point. And we will know the sizes of those segments that are involved. And we're going to make sure that, number one, our technology has the ability to detect all of those segments based on the resolution like Jenna was talking about earlier. And number two, we're going to make sure that, we look closely at these segments to identify these so that we don't, miss those specific parts.
Mm-hmm. Okay, excellent. Jenna, how accurate is PGT-SR testing?
It's about the same as PGT-A, mid to high 90s. But again, as Rachel just said, it's really important that we know beforehand what we're looking for, and we know that we can detect it. Obviously, if there's a rearrangement that's below the resolution of the test, it's not going to be an accurate way to test for that. And because like as Rachel said, we can sometimes test people on PGT-A who don't know that they carry a rearrangement, and then just by chance we can see lots of rearrangements in their embryos that are unbalanced, and then we can sort of infer that that they are a carrier of a structural rearrangement. Although that is true, some people look at that and say, oh, well, we don't need to review, we don't need to have the lab review the case beforehand because they'll pick up on it anyway. But again, it's really important that review process happen before to ensure that the technology, in fact, can pick it up. And if PGT-A cannot, as Rachel said, then we can try more of a PGT-M method.
To detect it. Okay, excellent. All right, and the last thing I want to talk about to two genetic counselors, I figure this is the perfect audience to our perfect panel to stress this, and that is the importance of genetic counseling. We are huge believers in making certain that whoever is doing your genetic testing, that they have genetic counselors available. And I'm just going to give, let's see, Rachel, for you, if you want to just give us a plug for the importance of genetic counselors to helping patients interpret their results.
Yeah, so, luckily, with with us at our lab, we do have a lot of genetic counselors that our role is to speak with our patients going through this testing process. And there's also an additional value even to outside genetic counselors as well, which we usually refer to them as clinical genetic counselors. So the counselors that are maybe in the IVF setting or the prenatal setting. So really, all of these options are great for the patient. Ultimately, we want to make sure that the patient is understanding the process that they're going through. What's their timeline going to look like, what they can expect overall from going through this. We also want to make sure that they are going to understand what their results mean, whether that's us talking to them before they're doing their testing so they know what to expect there, or sometimes we talk to them after their results just to help them understand them and make sure that they, are reading them correctly. And it's also really important in, talking with patients so that they understand, not only what are we looking for, but what are we not looking for in the testing, because one of the misconceptions that we get sometimes is, oh, well, you're testing all of the chromosomes, so you'll know everything. You'll know if my child might have autism or a birth defect. And so we also want to set those expectations there that, the testing is looking for what it's looking for, but we obviously can't look for everything, and we can't guarantee, a quote unquote normal or healthy baby at the end of this. But we have these genetic tests that can increase that chance for that successful outcome. And, sometimes there might be additional testing that's indicated, just to help identify that we really are doing all of the most optimal testing for the family. So the genetic counselor might be able to notice things by going through a family history or pregnancy history that might help with that. And then, of course, in the PGTM process, it's because it's so much more in detail. The genetic counselors are really kind of walking through that whole process with the patient. It's usually a multi-step process. So we're hopping on the phone with them a few times, potentially, emailing back and forth a bit to make sure all the steps are in place for that.
Yeah, excellent. Thank you so much, Rachel Cavey and Jenna Miller, for talking with us today about genetic testing. It is hugely important in IVF and becoming even more so as time goes on. To get more information about Rachel and Jenna, as well as Cooper Genomics, you can go to their website, and they have a lot of good information, educational information on their website. So I really encourage you to go there because it's a great place to learn. And that is coopergenomics.com. The views expressed in this show are those of the guests and do not necessarily reflect the position of creating a family, our partners, or our underwriters. And keep in mind that the information given in this interview is general advice. To understand how it applies to your specific situation, you need to work with your infertility professional. Thank you so much for joining us today, and I will see you next week.